Hello, hello, hello. Hello to all of you. I'm here. I'm back. I did not get a chance to stream last week, but I am glad to be here with you all right now. Whoa, this is way too big. One moment. Now that's way too small. How are you all doing? Let me know. I've been away for a little bit. Yes, what's up? Hello, John the Eosinophil. Hello, nothing. Welcome to the stream. It's on Big Fi right now. Let me get... Oh, that's why, because this is the right overlay. There we go. Hello to all of you. What's up? I want to hear, what do you all have going on while we file in and talk about Plague Inc. Hello, stupid channel. Hello. Mr. Neutrophil, I just wanted to say your channel reminds me of Sam Onilla Academy and Tirzu, and I really hope you blow up. Love the videos that I've watched from you. Thank you so much. Yeah, Tirzu was actually a huge inspiration for this channel, so I definitely would love to follow in their footsteps. Hello, General Mustache. Let's go. Indeed. John the Eosinophil, you're great. Thank you. You're great. Thank you. I'm glad to hear that you're great. I'm doing quite well. I'm happy to be streaming today. Work has been kind of rough, but I'm freaking back, y'all. Hello, Miguel Craft. Hello, welcome to the stream. Hello, Surprise Pants. Yes, Fi strong. Yeah. So I guess while people are kind of filing in, let's talk a little bit about Plague Inc. and sort of this whole adventure that I've had. I mean, I think I owe a lot of my current subscribers to the decision to cover Plague Inc. So I'll tell a little story about that while I get some lo-fi on. And please let me know if the music is too loud or if the music needs adjusting. Hello, Mitochondrian. Welcome. Hello. So, yeah, I mean, if you go through kind of the history of my channel, you know it started with covering video game biology, and I kind of at the beginning, you know, I did StarCraft, and then I didn't really know where to go from there. I knew I wanted to do Pokemon, but after I had done a couple of those Pokemon shorts, I found myself out of ideas, which was a bad sign considering how young of a channel this is, right? But then I thought, hey, why don't I cover Plague Inc? I bet that would put me through at least another 2 months worth of content. And I think that turned out to be a really good idea. And I think it's been such an interesting journey in the sense that, like, you know, as a content creator, you can't really tell what people will like until you just put stuff out there. Because I thought Pokemon biology, that's got to be where, you know, the biology enthusiasts are, because Pokemon is just so huge. But as I've come to realize, the real ones, y'all are one and the same as me, because when I was just a wee stem cell, I was a huge, like, I don't know, disease freak. If you've watched my rabies video, you already know that, like, nightly I would pray to avoid getting all these rare and horrible diseases. And I think Plague Inc. definitely taps into that kind of morbid curiosity, but also genuine interest for biology. So that's very exciting that I get to fully geek out at stuff like this. I have with me here a little book that I've gotten, so I don't have to keep on flipping between notes on my screen. So I want to make sure that before I properly get into the Plague Inc. tier list, that I want to get a couple of things straight when it comes to how this is going to go down, right? So I am a cell and molecular biologist, which means I am not a medical doctor or an epidemiologist, which means that I'm not, like, super familiar with how disease works. The thing about being a cell and molecular biologist is that our scope of expertise, you could say, happens at a much smaller scale than, say, a doctor would be familiar with. Well, doctors are familiar with the small scale too, but doctors are more concerned with the overall health of the individual, right? And so I can't promise that this will be rigorous in terms of disease, but I can tell you a couple of things about how individual little guys like bacteria work, which I think is going to be very exciting. So, the thing about Plague Inc. is that as a game about disease, it's actually really, really good. You might not get that idea from how harsh I've been in my YouTube shorts, but actually, if you look online, I believe the CDC actually endorsed Plague Inc. for being an excellent teaching tool for epidemiology. So teaching people how disease spreads, Plague Inc. is great. I couldn't take that away from Plague Inc. at all. If it's endorsed by the CDC, hey, it's good enough for me, right? Nothing. I kind of wanted to be an epidemiologist. Yeah, I think that looks correct in terms of the spelling. But yeah, I think epidemiology is super, super interesting. I don't quite have the rigor for that kind of research, though, although I do think it's very important and very cool. Yeah. What is your favorite plague in the Plague Inc. game? I will get to that on this tier list. We'll see that in just a little bit. So, the thing that is the focus of this Plague Inc. stream is going to be the mentions of actual biology. And so I'm defining that as, can the plagues in the list do these things molecularly? Is there a known mechanism by which these plagues actually can affect their symptoms and unique abilities, right? So I'm mostly going to be picking on the language of the game, and this might seem to be a little like too picky, but in reality, scientists have to be very picky when it comes to language, because if there's one thing that scientific writers hate is ambiguity. So if you think that I'm being overly mean to this game, you know, yeah, it's a game. It's not going to be perfect, but there are ways, and I hope to make my case to Endemic Studios. There are ways to change the language to make the game a lot more official, you know, sounding or more in line with current biology, which I think is going to be very exciting, right? All right. One thing to note is that I'm going to be saying the word precision like a lot today. I'm going to be talking a lot about the term precision. So what does precision actually mean? Precision means that, well, precision in writing means that if you use a certain term or phrases, then that phrase cannot be interpreted in multiple different ways. So if something's imprecise, that's another word or another way of saying that it's vague. And so these plagues are going to lose points if they are imprecise, if the language is too muddy, we'll say. Hello, Marshy. Welcome to the stream. Make a video about prions. So, you know, there have been a couple of Plague Inc. shorts about prions, but I am planning to make a big prion video over the summer, so stay subscribed for that. Okay, well, with that being said, let's get right freaking into it, okay? So, when I started to prepare for this stream, I started just by looking at the plagues, right? But then I realized that there's a lot more stuff in the game than just the natural plagues. And so I couldn't fit all of the plagues in, so you'll notice that we're missing, you know, Necrovirus and Nirax worm, you know, the fictional diseases. I'll probably get to those in a different stream because there's just so much to talk about with Vanilla Plague Inc. So let's start with ATP boost. So these up here in this first row, this is sort of the starting bonuses that you get in Plague Inc. So let me pull up exactly what ATP boost does. Bang, we got graphics out here. Look at us. All right. So ATP boost, which again is one of these starting bonuses, it gives you bonus DNA at the beginning. So what actually is ATP? ATP, and I'm sure Mitochondrion in chat can tell us, ATP is cellular fuel. So cellular fuel, it is a molecule that when coupled with other chemical reactions makes those chemical reactions easier to happen, basically. And so at first glance, there seems to be a bit of a disconnect between boosting this cellular fuel and acquiring extra DNA. And I get the fact that, like, just because you have more DNA doesn't necessarily mean you are more complex of an organism. If you don't believe me, there are vegetables with more DNA than you have in terms of base pairs. So I get that having more DNA is a game function, and that's fine with me. What I find to be not perfect is that the line between having more ATP and like having more, I don't know, mutational capacity is not necessarily all that clear. And so here's a figure that I drew up. This is like my, I guess I should also kind of explain this tier list, right? So up top, I have superb, right? I have like the tier where, yeah, the biology is solid. It makes a lot of sense. This tier, the I guess tier. The I guess tier is like, I could draw the line. It's not very strong. It's not a very direct route, but I could see the connection. Non sequitur basically means that the information in the game does not follow logically to its conclusion. So this is stuff that is like okay to say in biology, but has nothing to do with like the in-game representation. Then we have the technobabble tier, which is just words, doesn't really mean anything. And then we have the no tier, where the biology is actually just wrong. So that's how we're going to rank these, right? So ATP boost, where does this lie on this tier list? Rayamite says, perhaps the rushing of spending the ATP for reproduction causes imperfect reproduction, resulting in more mutations. That's not a bad hypothesis. I have something similar, actually, to what you came up with. So I'll share that in just a second. Okay, so let's see how good this will come out to be. So let's hide this. Let's just take a look at what ATP actually looks like. I did not think this through. So let's hide the tier list and take a look at the chemical structure of ATP real quick. Okay. So up here, this molecule, that is ATP. If you are not aware, ATP is, you could kind of think of it as used up during chemical reactions to make things happen in the cell. And so if you check out my mitochondria stream, you'll hear a little bit more about how ATP actually generates that energy. But the short end of it is that there are these phosphate groups. You'll see that there are a lot of like lines drawn between these. That's because there are a lot of electrons that are kind of creating these bonds. And so there's a large negative charge in between these molecules. And when you couple the breakdown of this molecule, the cleavage of these phosphate groups, that is energetically favorable and allows you to do other things and cause work to be done in the cell. So what does that have to do with DNA? Well, if we pull up the structure of DNA, so this is one side of the DNA ladder, and this is the other side of the DNA ladder, you'll notice that this structure here, this adenine, is the same adenine that's actually found in DNA, right? It's because ATP is actually one of the, we'll call it reactants to actually build DNA. So not only is ATP useful because it causes cellular reactions to happen, it's not the only molecule that does, but it's a major one. It's also a fundamental building block of DNA itself. So I propose that ATP boost is a little playing favorites, right? ATP is one of the DNA bases. What about thymine? What about GTP, right? There are other nucleobases. And so to make it really clear what this is about, I would propose to call ATP boost something like nucleotide boost, which is a lot more equal and fair to all the other nucleobases. So with that being said, where would I put ATP boost? I would put ATP boost in the I guess tier. Right? I guess. It's not great. I kind of had to draw the line together myself, but I could draw the line, and that's kind of what defines that tier. F. Yang, ATP is not a DNA nucleotide. Yeah, ATP is consumed when making DNA. Yeah. Okay, so yeah, ATP boost goes in the I guess tier. I could draw the line, but you know, it's not entirely clear. Let's see. What about in bacteria? Would it really count since it's one of the primary resources for something like a human cell? But in bacteria, I'm not so sure if they all have something similar. So ATP as cellular fuel is relatively universal. Bacteria can break down sugars to achieve the same effect in terms of generating ATP. So it's a relatively universal reactant when it comes to doing chemistry within the cell. Okay. All right, so the next on the list is metabolic jump. So let's pull that one up. So what does metabolic jump do? So that's not showing up, which is a little concerning. Where is the thing? Give me 1 second. I had all these graphics prepared. Where are they? OBS is freaking out. That still works. I wonder why these don't work. I might just read it off then, if the graphic doesn't really come up. It's a little sad, but, you know, we do what we can when OBS is being OBS. Yeah, it wouldn't be a real stream without some tech issues. True. I do have them all loaded, so I can just read them. It's just it's a little less fun to do that. Okay. All right. So, metabolic jump. Thanks for coming to the stream, mitochondria. Okay, so metabolic jump. So this the starting bonus gets you more DNA from popping red biohazard bubbles. So in my imagination, this is like, you know, getting or having your mutation rate kind of catalyzed by infection for in some capacity, right? Some sort of like the more this vector, the more this pathogen is like passed around, something about that process is causing it to mutate more quickly. And so metabolic jump doesn't really have anything to do with mutation. First of all, this is the first time I'm going to use the term imprecise, because what does jump even mean, right? Like, does it mean that it's skipping over something, like jumping over a barrier? Does it mean jump as in like an escalation, like you've increased metabolism? It's not entirely sure, so that gets the first imprecise mention. But I kind of figured that I could try to reword this, because as it stands, I'm not really able to figure out a way to connect metabolic jump with increased mutation rate, as it refers to with the red biohazard bubbles. So instead I propose something like transformation, DNA transformation, or genetic transformation, I should say. And to me, that's a little more real in the sense where bacteria can kind of take up DNA sequences from their environment. So that makes for a potential like way that you can kind of think about, you know, bacteria being exposed to other bacteria that are infectious or carrying infectious genes, and then sharing those infectious genes. So maybe that improves these like infectivity mutations. Something similar could, you know, with suspension of disbelief, happen in other organisms as well. But like that's as close as I could get to trying to fix or trying to make clear what metabolic jump would actually do. Raynight, more drastic mutations, or perhaps more mutations off an unstable DNA mutation could line up with ATP boost. That is true. The line is still a little not as direct as I would prefer it, right? Right. So we'll give metabolic jump a very middling grade, I think. And we'll get to the plagues. Don't worry. So this is just technobabble. Cytochrome surge, this is also going to be in the technobabble tier. If you look at what a cytochrome is, these are proteins that catalyze redox reactions. So they are involved in like metabolic pathways, not an obvious line between popping orange bubbles for mutations. Also, surge is imprecise because what is a surge in this case? Like it's upregulated, which is the term that biologists would typically use. Or activated is another possible way to determine or to describe whether or not this has activity or not. Yeah, cytochrome surge. This is another technobabble one. Catalytic switch. So this is you get DNA from cure bubbles. I mean, I get that it's a game, so the fact that there are scientists doing research on the pathogens, there's not really a line that you could draw between the fact that research is happening and like why that would affect global mutation rate of a pathogen. So this is also unfortunately technobabble, although it's an understandable kind of technobabble because that kind of mechanism might just not exist, right? Rheonite responding to tested cures, yeah, maybe like if they were cultivating antibiotic resistance in like laboratory tests. I guess I could bump that to the I guessed here. Yeah, that makes sense. Welcome, Thomas, the thermonuclear bomb. Metabolic jump, meaning it metabolizes faster, then it reproduces faster, and it mutates more with the increased reproduction. Yeah, okay. I could see that. And we're actually going to talk a little bit about that philosophy when we talk about the virus as a plague. So actually, I'll upgrade that to to the I guessed here. That's fine. That's fine. The kind of justification for what is it? Catalytic switch, like leading to increased mutation rate, would also depend on like all of those scientists totally failing at containing their pathogens, which could happen, but we would prefer that to happen as little as possible, but certainly I suppose possible. Okay, next is metabolic hijack. Which one was this? Metabolic hijack. Orange and red bubbles are popped automatically for you. So metabolic hijack. So I guess this would kind of be like an increase in speed of mutations. Very like vague. Again, with the metabolic thing, I can see the argument for metabolic jump, but metabolic hijack to me truly is like random technobabble, because whose metabolism are you hijacking? Why would that cause you to quote unquote generate mutations easier? I can't really see that line. And it's not really a term that we would use in, at least that I've seen. So I've just instead fixed it to genetic promiscuity. Not great either, but yeah, this one was a hard one to make work. So into technobabble it goes. All right. Hello, Rodeo Grill. Hello, welcome to the stream. Metabolic hijack, as in hijacking host cellular machinery. Maybe, but I'm not entirely sure what the line would be to generating mutations faster in the pathogen. Maybe. I'm open to the idea of bumping it up, but for now it lives in the technobabble tier. Okay. There are two quick ones here, Darwinist and Creationist. So Darwinist increases the chance of your plague mutating, so it's very easily understood. That's not really like a mechanism, but it's a nice fun nod, I guess, to, Darwin as the main point man, I suppose, of our modern understanding of evolution. So this one goes in the I guess tier. Similar with creationism. Because creationism makes things harder to mutate. Again, it's not really a direct line, but I like the illusion at least. Decreasing the existing requirement for mutation sounds like too big a maybe, though. I'm not entirely sure which one of those you're referring to, Rheonite. Decreasing the existing requirements for mutation. Yeah, I'm not entirely sure. Okay, so those are the Darwinist and creationist ones. Now we get to genetic mimicry. So which one is that? The one before that we were on. I see. So that one was metabolic jump. Metabolic hijack, I believe. Decreasing the existing requirements for mutations. Sounds like too big of a maybe. Yeah, I have tunnel vision on yeah, I don't quite remember. Sorry. Maybe a decrease in requirement for genetic shifts to occur. Okay, sorry. I didn't read the comment before. I see. Messing up DNA polymerase sometimes increase mutation, but that's not really a hijack. Yeah, I agree with Toxo. And we'll talk about fidelity in polymerases when we get to the virus one, but we'll keep that one in mind for sure. Okay, let's talk about genetic mimicry. Or genetic mimic, I think, is what the game uses. So this one just says your plague is harder to cure. So I actually like this one because genetic mimicry is something that pathogens are capable of. Sometimes they use it as a form of escape. So if you have a virus that can present an antigen that looks like something that belongs to you instead of something that belongs to the virus, that is one potential way for it to escape the immune system. It might make it harder to cure as well, as if the antigens that are being displayed by infected cells look like your own. That might make it harder for the development of, say, vaccines. So I actually really like genetic mimicry as one of these settings. So this is going to get the honor of the first superb category, which I really like. Maybe it copies cell protein markers, causing antibodies to not target them immediately. Yeah, exactly. So genetic mimicry, if we're looking, if an infected cell looks like a healthy cell to the immune system, then that would reasonably make it harder to cure if you're relying on your cure teaching the immune system which cells are actually infected. So I like genetic mimicry as a Plague Inc. thing. Yeah. Fire, superb, yes. Superb biology with genetic mimicry. Love that. Okay, base oxidation and base hydrolysis. So we'll get these up. It's such a shame that I can't actually show these because OBS is being OBS. I thought this just causes the adaptive immune system to recognize host proteins as foreign. I think it works the other way around, right? To recognize pathogen proteins as self, which I believe some viruses are capable of doing. There are, I believe for sure, not phosphomimics, but antigen mimics that some viruses can put on the cell surface to make it a little harder for the immune system to react to. Fun fact, the vampire game mode won't start a cure if you've never spread it, making the game much less time restricted. That's true. I mean, if we're talking about like which game modes I actually enjoy, I'd have to say that the vampire game mode is not one of my favorites. But we all play the game, I think, for different reasons, maybe. Okay. So where was I? Base hydrolysis and oxidation. Right. So base oxidation makes the plague easier to mutate transmissions, and base hydrolysis makes the plague able to mutate abilities. So these are both, you know, in that line of like stuff that makes it easier for your pathogen to mutate. And I think that these are actually, at first I was like, I don't know, seems like technobabble. But then I thought about it and I'm like, yeah, I kind of get it, because one of the types of damage that your DNA can endure, or that the DNA of a pathogen can endure, is oxidation. So if your DNA is, say, irradiated or, some kind of damage, you can end up with an oxidated base, and that oxidated base can then be repaired. But if that repair is imperfect, if the repair machinery for your DNA or the pathogen's DNA can't quite repair that correctly, then you can end up with a mutation. Similarly, for base hydrolysis, although I think that's not quite as clear to me that would result in a mutation, but base hydrolysis is the mechanism by which enzymes like DNases cut DNA. So it's another form of, you could call it damaging DNA. And so because you can draw a line between base oxidation, base hydrolysis, and mutation, I'll put it in, I don't want to put it in the superb tier because I think I have a couple that are reserved for that high of an honor. So I'll put them in the I guess tier. What if it could mess with telomerase and restrict corrective DNA repair? Yeah, so what would you call that though? Because it's got to be like a snappy name, right, to fit inside the flavor text of Plague Inc. Because some of these that I wanted to fix, I wanted to like rename. So, but keep it with the biological mysticism almost, which is fun. Ionized helix. I love this one actually. I love this one. Maybe not for the best reasons, but ionized helix. Let's see. I don't even remember what this does. I just love the name so much. What does ionized helix do? No, did I not save this as a picture? Let me look it up real quick. Ionized helix, Plague Inc. Whoa. Let's get back to the right thing. There we go. So ionized helix, this gene grants bonus DNA when devolving. So this is another one where it's a game, and so there's not really a devolving mechanism, right? You can't really devolve. Evolution goes in one direction, and it either goes or it stops upon extinction, right? But that's okay. Like, it's a game. I get it. I want to focus on the name ionized helix, though, because this is something that we actually touched upon when we talked about ATP boost. So I really hope I can bring up that image again. I can. I don't know why these other ones won't show up, but that's really sad. Hold on. I'm going to try something. Let's take a look at this DNA first. And we'll blow this up. I think maybe it's just not like loading on my OBS correctly, but we'll make this real big. And hopefully, it's kind of hard to see. No, it's hard to see. Okay, that's okay. That's all right. That's all right. We'll deal. So the reason why I like ionized helix as one of these bonuses is because DNA, if it's in solution, if it's in water and it's happy, it will carry a charge. It will be ionized, right? There are these phosphates that, when deprotonated, will carry a charge, meaning that the backbone of DNA is already ionized. It's just kind of a property of DNA to begin with. And so I guess for anything, like if it's a game mechanic that doesn't make sense, it might as well be something that's just put out there, because I certainly couldn't come up with a better name for biology that doesn't exist. So this gets the non sequitur. The helix of DNA certainly is ionized, but it kind of doesn't really mean anything. It's just kind of one of these properties of DNA. So into non sequitur it goes. Right. So next up, translesion. So this is a term that I actually was not familiar with. Again, this didn't seem to load, so let's look this up real quick. Translesion, Plague Inc. Yapping is a trait that will come in handy if you are a science communicator. We love yapping. Translesion. So translesion, the effect is costs to devolve don't increase. And so, yeah, again, it's another mechanism that doesn't really exist in biology, so we'll give it a pass for that. But I actually had never heard of the term translesion. And translesion is a term that actually exists, and it's a type of DNA repair in response to a very certain DNA injury. So I'm actually going to put it right next to ionized helix just because it's not attached to anything that makes sense in game, but it is a real thing, so kudos to that. I think that's kind of fun. And I actually learned something because, yeah, something that I wasn't aware of. Alrighty. So, yes, thank you. I still have the picture of DNA up. My bad. Let's put back the tier list. Tier list, great. Yes, so I put translesion right next to ionized helix because they're kind of in the same realm of like they are real things, but they don't really mean anything with regard to the game. Like there's no tangible connection, which I think is fine. Hello, Aeon. Welcome to the stream. Welcome. Hello, Malik. Welcome, welcome. Thank you for pointing out that when I talk on stream, I'm a terrible streamer in this regard. I get so lost in the sauce of what I'm talking about that I'm not really watching the second monitor. Thank you for pointing it out. Yeah, translesion is also an adjective, not a noun. True. I didn't have too much time to read into what translesion was, but once I saw that it was a real thing, I was like, oh, cool. Okay, so not technobabble. But definitely interesting, and I might take a further look into it in the future. So, with those out of the way, we can finally get into the actual plagues themselves. I really want to show these because there's like a lot to read. So I'm going to try reloading these assets in. And in the meantime, I'm going to throw up a poll because I'm kind of curious as to what your favorite, let's see, I need to open my stream because I'm not monitoring it right now. But I kind of want to know like what your favorite plague. Yeah. Let me know what your favorite plague in game to play is, not necessarily your favorite one to think about, but what your favorite game to play is. In the meantime, I'm going to try reloading these assets into the game because into the stream because I really want to show these off. Please work. Okay. Let's try that. So they definitely appeared on screen all at once. Great, great, great. Hopefully we'll be able to inspect these because this is what y'all probably came here for. I had my fun talking about the setups, the starting bonuses, but I think what people really want are the actual plagues themselves, which I also want to talk about. All right. There aren't actually, annoyingly enough, YouTube only allows you 4 options on the poll, and there are way more than 4 plagues, so I just ended up retracting my offer to poll. All right. Now I was here for the starting bonuses because aligning Technobabble to something tangible is always cool because you learn, yeah, I love Plague Inc. the game. Like, I think it's a great game. I just think that the Technobabble is a little goofy sometimes. It's awesome. My streamer is a trickster. Unintentionally so. I am kowtowing. I apologize. My favorite are prions. Prions are cool. Yes, prions are super cool. One of the things I used to pray about back when I was praying was that I would never get a prion disease because, you know, when I was a kid, that's when mad cow happened, and that was super scary. We won't talk about mad cow today, but we are going to get to prions. I'm always torn on bacteria and viruses when the fun to think about topic is brought up. Yeah, I love bacteria and viruses. They're both capable of doing some amazing, amazing things. Do multiple polls and then have that be elimination round for final poll. Yeah, we could try that. This might build a little hype. Build a little hype for actually talking about these guys. So I'll do the first four: bacteria, virus, fungi, and parasite. Pathogen tournament. Let's go. Let's pull it up. Virus feels fun with the RNG aspect. Watching mutation after mutation happen is just funny. Yeah, I had like an ambition to play viruses without touching the keyboard, just like load it up and see if the virus itself could mutate itself into a winning virus. I think I've tried it like for 4 streams and it didn't really pan out, and they were kind of awkward streams to do. But I also find virus to be very, very fun. What is my favorite disease? There are so many interesting ones that I don't have a great answer for that quite yet. There are some that are very near and dear to my heart in the sense that they were closely related to the research area that I had studied for grad school. They're not anything that I covered so far, but I don't really have a single favorite, especially because when making content for the channel, I tend to bounce because if I choose something to cover, it's because I find it insanely interesting in that moment. And so I don't know, there are a lot that are really cool. Name one. I'm partial to tuberculosis, I think, as a immune evader. They're so tenacious, and the mechanisms by which they evade the immune system are so clever. Tuberculosis was actually one of the inspirations for this channel as well, in terms of what direction I would take from it, because when I heard the story of tuberculosis, I was like, I gotta share this. Like, no one, I shouldn't say nobody, but there are not many popular science, creators talking about molecular mechanism of disease. I mean, Kierkegaard is doing it, but at the time that I heard about tuberculosis, Kierkegaard hadn't done it. But yeah, if I had to say one that is in my favorites, it would probably be tuberculosis. For evasion of the immune system, I'm stuck between rabies and tuberculosis. Exactly. That's why I did them as videos first, because I was so excited to. I couldn't really hold off. Okay. I kind of want to check the results of this poll, and then we'll get into talking about bacteria. All right, viruses. Okay. We'll keep this poll going up until you know what? Yeah, I think that'd be kind of fun. I'll do bacteria, virus, fungi, and parasite, and then I'll do round 2 of the pathogen poll. I think that'd be fun. Okay, so let's talk a little bit about bacteria, and hopefully I can pull up their actual block. Yes, haha, OBS. But now you won't let me move this thing. Okay, great. We're doing it. We fixed it. Okay, let's talk about bacterial resistance. So, in the sort of mechanism of the game mechanics, a lot of the pathogens share a lot of stuff in common. So we're actually just going to look at stuff that is unique to each pathogen, at least in the vanilla Plague Inc. game without the DLC or without the extra special plagues. So the unique part about bacteria, their special ability, is this thing called bacterial resilience. I actually like the term resilience because it's not at least to my knowledge, it's not really a term that people use. There's bacterial resistance, or rather antibiotic resistance, which is, you put on antibiotic and bacteria are capable of evolving ways to deal with those antibiotics. You also have bacterial persistence, which not a lot of people are familiar with, but basically, if you try to destroy a population of bacteria in various ways, a proportion of those bacteria are actually capable of reverting to sort of like a stasis. Like they can shut down a lot of their own metabolism and kind of hole up and protect themselves from being destroyed. So resilience kind of squeezes into this like unknown territory, which I think is kind of fun because this is not typically, I would say, because I'm not an epidemiologist, so I'm not too familiar with this aspect of bacterial biology, but I don't often hear about membrane integrity when it comes to disease. So if we look at bacterial resistance as a trait, we'll see that on the first stage, we create a bacteria shell to protect against all climates. And then when you level up, bacterial shell gets thickened, further increasing bacterial survivability in climates. And then in the third stage, bacterial shell practically impregnable, furthering increasing bacterial survivability in all climates. So in terms of game mechanics, this is basically saying that the bacteria are better at surviving hot and cold because of their quote unquote bacterial shell. Yeah, the easiest and simplest in the game. Yep, that's correct, nothing. It's a very tutorial level plague. It's the first one that you generally use. Not like immunity, correct. Resilience and persistence are not really immunity. I don't really know too much about Hep D, actually, or the hepatitis viruses themselves. I think that'd be fun to do as a video one day. I bet they have some pretty nasty immune evasion stuff. Yeah, so the thing that I take issue with in the bacterial resilience block is this term bacterial shell. So this is another instance of imprecision. It's not very precise because bacteria have a cell wall and a cell membrane. You could refer to them as a bacterial envelope and people will know what you're talking about, but the term bacterial shell doesn't like really exist, right? So it gets some points docked for that, but also the idea that a thicker membrane results in more resilience to climate is also somewhat flawed. While there are aspects of membrane chemistry and physics that can make it that can make an organism more likely to survive cold and hot climates, it's not like unidirectional, right? Like just because you have a quote unquote thicker, more impenetrable bacterial shell slash membrane slash wall doesn't necessarily make you better at surviving hot climates. When in reality, bacteria and generally cell life in general, you want to maintain a level of flexibility. You don't want your membranes to be too, wibbly wobbly because you'll end up just disintegrating if your membranes can't hold themselves together. But if your membrane can't move, if you have no membrane flexibility, then that's really bad too. You need some level of flexibility. So it shouldn't really be like this idea that, oh, the thicker the membrane gets, the better. It should be stepwise, like maybe you have like the first stage be bacteria synthesize cholesterol molecules that maintain flexibility in all climates, or unique lipids in the membrane that are capable of adjusting flexibility. So it should be about flexibility, not rigidity. Also, this idea that it would be beneficial for a bacteria to have something described as an impregnable wall is very questionable. And again, impregnable, also another very imprecise term. What are you impenetrable, like impregnable to? Water? That wouldn't be good, right? Bacteria need water. So what does impregnable mean? It's just not a very disciplined way of describing bacteria. So let me catch up on chat while I was on my rant. What if a bioweapon could invert hydrophobic and hydrophilic properties in a cell, forcing their membranes to turn inside out and rupture? Interestingly enough, that would be very, very difficult to do electrochemically. Membranes are the way that they are because of physics and chemistry. the electrical forces that keep your membrane together, they're not. It's kind of hard to describe, but physics is keeping your membrane in place. It would be pretty wild to be able to do that. Although the closest thing that I can think of is just soap. Soap will destroy membranes because soap is very good at binding hydrophobic and hydrophilic parts and breaking those lipids up. Wayward Planet, Plague Inc. is inaccurate. Next, you'll tell me vampires don't exist. I mean, yeah, at the top of the stream, I was like, it doesn't have to be accurate. It's just a fun way to talk about biology. But I'm also heartbroken that vampires don't exist. Team Edward. A thicker membrane just makes it vulnerable to temperature shock when flipping between temperatures. Yeah, you want flexibility in your membrane. Yes, yes, yes. I would not think the extremophile bacteria survive using a thick serial. Exactly, exactly. So it's kind of this like very simple way of thinking about the membrane, one that isn't entirely representative of reality, we'll say. Aeon, you can tell because that's the way it is. I'm not entirely sure what that's referring to. Vibrio. Vibrio is a great bacteria to cover one day. I would love to cover Vibrio. Soap literally dissolves bacteria. Yep. Soap is pretty gnarly, which is why your skin is an MVP organ, because your skin protects all of your vulnerable cells from things like soap as well. Also, welcome to the stream, more juice. They should have looked into the disordered proteins in the tardigrades for inspiration for extremophiles. True. Thank you, Toxo, for plugging my video about tardigrades. You should go watch that if you haven't. All right, anyway, so enough about bacteria. So you kind of get my gist, right? Like, it's cool. It refers to real biology. It's fine, but it's not really accurate, but it doesn't, like, it's not at the level of technobabble, right? Like, it attempts. So it gets an I guess tier. Okay. Next up, we have viruses. Okay. This one's another really fun one to talk about. I love this one. If I can only drag it correctly. Come on, OBS. Yes, thank you, OBS. Thank you. Viral instability. Okay. So, we got a lot of imprecision in this description. So the first stage of this viral instability is that the DNA becomes unstable, increasing random mutations. So instability is like not a well-defined term when it comes to the context of DNA. At least I haven't really seen instability being used in papers talking about mutation. Maybe it's not very common, but I'm willing to give that a pass. But at the second stage, DNA occasionally fractures, increasing random mutations. This one's, it's a little... when DNA fractures, we usually call that a break, like a double-stranded break if we're talking about DNA, right? There are... When DNA breaks, it needs generally repairing because there are enzymes in organisms that repair DNA that is broken. And so if it repairs the DNA in a way that is incorrect, then that could generate mutations. So that's also like okay, although they don't really fill in the blank when it comes to then it has to be repaired, meaning that it then can be mutated. And then the third stage, DNA becomes highly erratic and fragmented, increasing random mutations. So erratic is not really a term that I've ever seen used to describe DNA. And fragmented is not a good thing for the virus, right? So by the language, and Yang, F. Yang, you're making a good point about mutations, but I would say that to give the game a pass and also, like, to me, it's fine. RNA viruses do have a higher rate of mutation than DNA viruses, but if Plague Inc. wants their plague to be a DNA virus, I'm all for that. What I'm not really for is the use of imprecise terms like fracture and erratic. And yeah, if your DNA is actually fragmented, like you broke the dang thing, that's not good for whatever organism or entity in the case of a virus. So that's kind of in the technobabble tier, I would say. As a grad student in virology, this makes me want to die. Yeah, it's so I'm not a virologist. So yeah, actually your comment earlier, I think on the original Plague Inc. Short was insightful. Although I'm probably more willing to give them a pass because it's not like DNA viruses don't mutate. It's just that RNA viruses are mutate more often. So I'm judging it maybe a bit more generously, I think. Biology is so interesting. It definitely is. Aren't mutations because transcriptase done goofed? So mutations can happen from a variety of different mechanisms. You can have, attempts at, so first of all, you can have damage, right? Damage causes mutations, damage from UV, damage from superoxides, oxidative species in or surrounding the DNA that can affect the DNA in the cell. And then when that DNA then has to be repaired, if it's repaired with the correct base, then you don't get a mutation. If it doesn't get repaired correctly, then you get a mutation. So you can have errors in polymerase where it's just bad at copying the DNA and it screws up putting the base that's supposed to be there. That's a possibility that can happen. But there are all sorts of different ways that mutations can occur. Yeah. So instead of instability, it could say enzyme dysregulation, something like that. Yeah, that was actually my original pitch, is that if you say something like low fidelity polymerase, low fidelity meaning that it is not very accurate, then that could result in erroneous copying of DNA. That's one possibility. Recombination too, excellent point. Yes, so recombination can lead to changes in subsequent DNA sequences as well. To be fair, without polymerase effing up once in a while, we wouldn't have biodiversity. Haha. Yes, exactly. So I was reading an interesting paper, kind of inspired by when F. Yang had pointed out the difference between RNA and DNA viruses, sort of whether or not high mutation rate is always increases fitness. So fitness in the evolutionary sense is, I'm not an evolutionary biologist, but from my understanding, it's the ability to pass on your DNA to the next generation. And there's like sweet spots from what I read. Like as a virus, mutation is not the worst thing, but if you get too high, then it's not good for you either. So viruses have sort of optimized their own mutation rate for whatever circumstances they find themselves in, which I think is pretty interesting. Endemic is supporting RNA virus erasure. True. Very true. Okay, so yeah, viral instability. Yeah, I'm gonna have to say that viral instability, the virus, unfortunately, I'm gonna have to put this into technobabble tier. Yeah. Okay. Fungus. Let's go fungus. Fungus Among Us. Always have to do this dance with OBS. Come on. Come on, come on. Before I move on, I'll read chat a little bit. Fitness is wherein microbes are swole as AF. True, true. Can we get some 5 strongs in the chat? Oh, I forgot to put I forgot to add. Oh, we have another emote that I forgot to put into chat. I'm so devastated. But yeah, swole microbes. True. Swolecules. True. Fungus is a cool one. I struggle a lot with it, but now it's easy now. True. I love this one. I unironically love this one. This one's great. I know. I'm sorry. Toxo. I got wrapped up in preparing for the stream and I forgot to add the emote. I'll do it right after the stream. So, in the fungus game mode, right, their main mechanism is to make use of these spore bursts. So they spread very slowly on their own in terms of fungal infection. But you have these like active abilities that causes clouds of spores to travel great distances so that you can infect countries that are geographically difficult to get to. And at first I was like, there's no way that fungal spores can travel like that far, right? Like there's no way that that's like a thing. And like, again, not an epidemiologist, so from what I understand generally, fungal infections are passed through close contact between species and not through fungal bursts, right? I think that's how they work, but I'm not 100% sure, not a doctor. But there's pretty good evidence that there was a fungal burst that started in Africa that infected coral reef in South America. If you've watched my fungus video short, which is absolutely bonkers. I don't know whether or not that could ever happen for transmission in humans, but the fact that fungi are capable of making spores so hardy and so light that they can be floated onto the wind, and so rigorous that they can not only survive, the blistering dry winds, but also the salty conditions of the ocean, is just insanely mind-blowing. Like, that's incredible. And since I'm not like a mycologist, like, because I was set on being like, no, this can't happen. Fungal infections are usually spread through like skin contact. So when I learned something that overturns a previous conception that I had, I think those are the most fun of things to learn. I think that would be really, really, that's like really, really cool. Are there any fungal illnesses IRL? Yeah, so like oh god, what is it called? Toxo, you might have to help me out with a Google search or something, but I think like foot and mouth disease. I can't remember. But yeah, there are for sure like fungal infections, IRL, yeast infections as well. How do I use channel emotes? So channel emotes are emotes that are reserved for members, which you can start by subscribing at $1 a month. I listened to a podcast on lung fungal infections. I'm not going to attempt to say that word. It sounded like a real unpleasant experience. Yeah, fungal infections, I think in the realm of immunology, are interesting. I'm not ready to talk about any fungal infections in depth for like a video, because fungi are pretty far from where I was trained. But the fact that fungi are much closer to our biology than, say, a virus or a bacteria, I imagine makes it a lot harder for the immune system to clear, which I think is interesting. Literal shrooms growing in your lungs. Fungi are really bizarre. I don't think that they would take the form of shrooms, because shrooms are fruiting bodies. But definitely, it would be fun to do one of these fungal infections for a long Find the Neutrophil video. Hand and foot disease is viral. Thank you so much. Yeast infections, thank you. It just so shows you how much I know about fungal infections, which is not a lot at all. Foot and mouth disease is endemic amongst the political class. Nice. Get them, more juice. Cordyceps in humans? I don't know that the cordyceps would have a evolutionary advantage to infecting humans at this point. Probably not too much to worry about. if I remember correctly, it was described as a bunch of microscopic mycelium tendrils in the alveoli. That's probably what I imagine too. I can't imagine a fruiting body actually happening inside of the body. I have no good reason why, but it would just seem weird to me. Yeah, so because of how, like, the fact that I like learned something brand new about biology and I was like flabbergasted, the fungi is actually going to get a superb rating joining genetic mimicry. I can't overstate how like because I actually wrote the entire fungi script for that short intending for it to be ridiculous. And then when I like dug into it and really like went hard in a Google Scholar, I was like, oh my god, I'm wrong. Thank God I like didn't hit the publish button or didn't like do the editing at that point because I was wrong. That's all. All right. Next we have Parasite. Parasite. Let's bring that up. Parasite. So the parasite's unique ability is called symbiosis. Here's another fun one, I think. So in symbiosis, we have the first stage where the parasite becomes more in tune with the host, making it less likely to be noticed. So again, in tune is scientifically imprecise. It doesn't really have a direct scientific meaning. And if you watched my Parasite short, you would know how I feel about that. But I'm willing to give it a pass and just hand wave it as some form of immune evasion. Second stage, Parasite fuses with the host DNA, making it less likely to be noticed and more likely to be transmitted. So I had to like take a break here when I was making the Parasite video because I felt bad for how crazy this is. Fuses with the host DNA. I was like, I don't want to like dunk on Endemic if the company isn't English speaking and this was some horrible mistranslation. But Endemic is an English company. They're based in the UK. So yeah, that's just nuts. Yeah, hookworms sleeping in the DNA ladder. They're not the same scale. And the picture, okay, so I guess I should back up and say, like, the other thing that I'm not in love with the parasite plague is the fact they're just called parasites, but a parasite isn't a species or a class or an order or anything like that. Parasites just describe the relationship of an organism to its host or its symbiotic partner, I guess you could say. What this is showing is something more like a helminth, like a worm, like an actual animal. And so the word parasite is again imprecise. They're showing a worm. I'm guessing they mean a worm, so why not just use the scientific name for a worm? But the fact that, you know, stage 2 is fusing with the DNA ladder is just bonkers. It's truly just nuts. So they definitely can't like get a pass on that. There's no way that I can make that fit. Yeah, exactly. How does a parasite fuse with DNA? That's a little technobabble. Exactly. That's actually technobabble. Oh god, a parasite is slithering in between my nucleotides. True. Not to mention that like parasites can be on helminths, I should say, can be on the scale of not micrometers, but millimeters. Like you can see a parasite, and DNA is at the angstrom, 10 to the minus 9, no, 10 to the minus 10 meters. Like incomprehensible technobabble. That one's terrible. And then stage 3, parasite infiltrates host brain, making it less likely to be noticed and more likely to be transmitted. I mean, sure, if you have an infection in your brain, that would probably make it more noticeable. Generally, when things that are in the brain that are not supposed to be in the brain are going to cause you to not have a good day. So I don't really buy that. That's not something that I agree with. But yeah, I mean, for stage 2 alone, the helminth parasite, it's going straight to technobabble tier. straight into Technobabble tier. And with that, I think we will conclude the poll. Let's see. Your favorite plague is the virus by just a little bit. That's so interesting. Actually, bacteria got a lot more love than I expected it to. Very good. Yeah. So let's get rid of this symbiosis. Oh, come on, OBS, please. Nice. All right. So the data are back. Favorite plague in round 1 is virus. I'll set up a second poll. Let's talk about the rest of the plagues. Fan favorite, I imagine. Nanovirus. And we'll do bioweapon. Are they saying people eat each other's brains? I don't know. It's oh no. I actually made this entire tier of no just for the Parasite, actually. Yeah, no, it's not even Technobabble. It's just not how biology works. Technobabble is just nonsense. The no tier is just not, it's just not possible. And there's very little that I would say is not possible in biology. That's another thing, right? There's stuff that can't be confirmed because it hasn't been observed in our natural world. And then there's like implying that a macroscopic parasite can fit in between DNA bases. Like that's just not physically possible. So this tier exists specifically for the Parasite to fit in. It's really, really that nuts. If I can get a Parasite to fuse with my DNA, is that how I become a superhero? I did a video for a I actually don't want to talk about that. Never mind. my dark past of other science communication ventures, we'll say. Let's say I have strong opinions on Spider-Man. My doc told me to cut back on the cholesterol. It's true. I believe brain is not the most healthy of food choices, although I would never do so just for fear of prion disease, which I'm sure you are all aware of, having tuned into a Plague Inc. stream. Stage 3 is sort of accurate, however Stage 2 is way too inaccurate. Tapeworms are visible to the human eye, so I don't see how they'd be able to squeeze into DNA unless they mean by take DNA and modify. See, that I think would have been like a better way to rule this out, right? Because we know that like organisms can pick up DNA from other sources, given a lot of that is mediated through bacteria and viruses and not really at the level of a helminth. For example, we have viral genes that have been inserted into us from ancient viruses. So you can have like DNA being picked up in the environment. Whether or not a worm can do it, I don't know. Maybe if there was a viral intermediate, maybe. But yeah, but it clearly says fuses with host DNA. That's inexcusable. I'm gonna do it in 2 seeds, so we'll do the first 3 and then the next 4 and then the next 3. So I'm like behind in chat because I'm slow. Bioweapon is a weird one for me. I worked in an organization that was designed to respond to biological WMDs, and they like all fell into viral or bacterial buckets. That's so interesting. I'm guessing you can't speak more of that because that sounds like sensitive information. But yeah, I can imagine, because in terms of spread, I mean, Plague Inc., I think, to their credit, I think got that right. If we think about the great plagues of human history, I can't name a single like great worm plague or an amoeba plague, but there are quite a few great plagues of bacteria and viruses. So that makes sense. Parasite in the Brain reminded me of the gnarly House MD episode where the tapeworm went too far. Ugh. Yeah, as a kid, as again, like a weird weird disease kid, I remember not being able to sleep when I discovered what bot flies are. If you want to value your sanity, don't Google that. I'm afraid of prions. Yeah, prions are definitely scary, but I think that we still have a good amount of time before they become apocalyptic, we'll say. And I really want to make that prion video, because every time you search prion content online, it's always like doom and gloom, because, you know, they're scary, like reasonably so. But I want to take my own angle and really talk up the research into prions, so I hope you'll stick around for that. Prions are underrated. Nobody cares about them until it becomes a problem. My comment section definitely does not think that prions are underrated. I can't tell you how many comments I've had in my various videos asking me to cover prions, which I will. I definitely will, but I want to do like a... I don't know. How does chat think about me doing... Because usually I do like really tight, in my opinion, tight stories, like really crisp stories about these pathogens. But for the prion video, I've been playing with the idea of literally doing all of the research that I can and listing everything that I know about prions. So I think maybe after this poll, we'll talk about what you would like to see from me in terms of how I cover prions. I mean, COVID did quote unquote well. Yeah, I mean, from COVID's point of view. Is it just me, or the bioweapon sort of similar to the virus except more lethal? We'll get into that, I think. Okay. So, let's talk about prions. We know them, we love them. I don't really know if my particular audience needs an intro to prions, but I'll talk about them anyway. Sorry, this is the wrong slide. This one, right? Here we go. Okay. Let's move me a little. You know what? I should probably have me above all of this. There we go. Okay. So prions. So for those of you who don't know, prions are proteins that have canonical function in the body, although I don't quite remember what they do now. They're a protein that I think is hypothesized to have functions in neurons. And I think that they help with the structure and connectivity of synapses, so the junction point between neurons. The issue, and what we know prions for nowadays, is the fact that once prions are misfolded, they can cause problems. So one of the problems that they can cause is protein aggregation. So once a prion misfolds, they are capable of causing other prions to misfold. And when you have enough of these misfolded prions, you get what's known as a protein aggregate. And these protein aggregates can be a real headache for neurons. So when cells are working well in your general day-to-day life, when there is sort of protein misfolding, you have these specialized proteins called chaperones that help them refold into the correct shape so that they are chemically very happy. And failing that, there are enzymes like the proteasome. Well, I guess the proteasome is more like a complex, but there are entities like the proteasome that can then destroy those aggregates in various ways. And so the reason why this is important is if you get too much of this unfolded protein, it creates a very toxic, unfriendly environment within the cell, which is bad news for something like a neuron. Doubly bad when you consider the fact that there is very limited neurogenesis in the brain, meaning that the brain doesn't tend to replenish neurons outside of a very select few populations. So when you lose it, you more or less lose it for good. And I know that there are a lot of comments on my rabies video that say, hey, you know, neurons do grow back. Not all of them. There are certain populations which have been shown to do it a little bit, certainly not enough to overcome a prion disease. So, with that in mind, these diseases, these prion diseases, do occur quite slowly, and that is accurately reflected in the prion gameplay. Like, it starts really, really slow, and it is really, really deadly to have one of these prion diseases. And it is really hard to spread. Like, once mad cow disease like hit the scene, like the world shut down consumption of beef. There was an enormous reaction by world governments to clamp that down because of what could have happened. And thankfully, we are better off for it today. And yeah, this class of disease would always give me nightmares when I was a kid, because it's like, once you have it, you don't know if you have it. You'll spend a couple, maybe up to a couple years of your life not knowing you have it, and then you just slowly lose it, which is devastating. But again, I think that there is reason to believe that there is hope for the future. It's not all doom and gloom. And I think F. Yang had brought up earlier in the comments that as the field maybe looks more favorably upon looking at Alzheimer's like a prion-like disease, there's going to be more attention to developing treatments for this kind of thing, which is exciting. That being said, there's a bit of wasted opportunity and a bit of inaccuracy when it comes to the prion mode as is, in terms of if we're not talking about the mad cow part, right? Because while the other plagues so far could get away because there is human-human transmission, as far as I know, in the rare event of, like, there's not really human-human transmission of prion diseases, save like Kuru, for example. Like, it would not act the same way. And there's also like data showing that you don't, I want to save it mostly for the prion video, just because I don't want to get anything wrong here. But basically, the mad cow scenario looks a lot more like a prion disease, or at least a game version of a prion disease, than the actual prion disease plague does in the standard version. That being said, we can take a little look at the neuroscience in here. In the first stage, we have neural breakdown, causes breakdown in concentration. I mean, this is just like, you know, C- for repeating the word breakdown twice. And also, breakdown is not precise. Like, what do you mean breakdown? Do you mean membrane damage? Do you mean apoptosis, cell death? Like, what does breakdown mean? It's not really scientifically defined. But fine, I can excuse that. Axonal swelling causes disorientation and intense confusion. So axonal swelling is a marker of brain damage. So, you know, that's not bad. And then cell death results in severe short-term memory loss, making complex tasks significantly harder. I think this is fine. I think, apart from the fact that prions don't really spread in the same way that other pathogens do, the prion accuracy, with the caveat that this is, of course, a game, relatively okay. I'm okay with this. Prion can get an I guess, goes in I guess tier. I think I'm pretty happy with that. Yeah, prions are wild. As far as I know, there's only one prion protein, but it can cause a variety of different diseases and symptoms. That's true. Depending on what type of prion you have, what version of the prion you have, where you got it from, it can manifest itself in several different ways. Again, if you want to sleep soundly tonight, I would not recommend looking into it any further until, of course, you watch my eventual video that comes out hopefully sometime during this summer. I have a hope that if prions actually become a problem, humanity can deal with it. Exactly. Like, I don't want to leave people off with like a total bummer, because I don't want to contribute to people's neuroses about disease. That's why, like, even though rabies is so horrible, if you get bit by an animal, go get a vaccine, you'll be fine. Like, it's one of the most efficacious treatments, I think, in all of history, right? Like, it's a good story in that way. Tuberculosis less so, but there's hope, because, you know, we were continuing to advance the scientific field, and we might live until the day where we can finally put that kind of great plague to its actual end. It certainly would be quite exciting. So I'm reading chat, catching up. I hope this is prion in pork, but not chicken. Well, as far as we know, well, no, not gonna say it. Not gonna say it. Leave it for the prion video. I don't want to talk too much about this. Leave it for the prion video. Okay. Third stage, I believe, is inaccurate. Cell death doesn't mean memory loss unless they... it's fine with just saying cells do have context and sort of inaccurate. That's fair. I think for a game, I still consider that to be acceptable. I think it's an acceptable line to draw. Like, I don't have to think too hard about connecting those dots, so I think it's okay. I see what you mean. Thank you, Toxic, for clarifying that. So you mean neurons in the brain. Yep, that makes sense. Central nervous neurons, maybe. Okay. Yeah. So, prion disease. I think that this is all right. Maybe not the best. I definitely would not lump it in with, my favorites. So it gets an I guess tier. Let's remove this. Yeah, I already put it in there, actually. Next up is the nanovirus, which is also a lot of fun to talk about. So... Okay, so the nanovirus, right? So this is a man-made virus, and there's not a whole lot to talk about it. Because just look at what these abilities are. Radical element stabilized, encryption breached, broadcast interceptor overload. It's like I'm playing StarCraft. Like, this isn't really biology. I mean, you could argue that it is, but it's not really biology based in any organism that kind of exists. I think the best that I tried to make it work was drawing a parallel to optogenetics, which is a technology that can use light to turn on and off genes. Not that that would really work for something like this, but scientists now can, in their model systems, when they're studying genes and they're studying model organisms, can do things like turn on and off genes with light. It's very specific. The light has to get into the cell. And so it's not like the best parallel for the mechanism by which we would talk about the nanovirus. So this is just like not biology, which is fine. Like, again, it's a game. I'm not mad about it, but this definitely just like has to go in the technobabble tier just because it's technobabble. I'm actually working on a science fiction prion scenario. I'm just stuck because of a bug in the government actions. Other than that, I finished the symptoms, abilities, and other stuff. Hopefully I'll reach it somewhere. That sounds really cool. I hope that my video comes out before you actually finish, if you need ideas for... Well, I don't know if you want a happy ending or not. Because there's some compelling research out there, I think, when it comes to prion stuff that I want to get into. What is my favorite kind of white blood cell? I mean, What do you think? Is this like an engineered nanotech robot that somehow went awry? Yes, that is what the nanovirus is. And so the gameplay mode basically has you, as humanity, try to sorry, that's cure mode. But basically you're trying to evolve ? But the way that the cure works in this plague mode is that they're sending like signals to try to like jam the nanovirus. They're like trying to broadcast like a kill switch to get all the viruses to shut down. Sorry, that's a good question. I should have explained that at the top of the nanovirus segment. If I were to play Plague Inc., I would name my nanovirus Iron Man because of the nanosuit. That makes a lot of sense to me. Does make a lot of sense. I'm just joking that the nanovirus is so ridiculous that it's practically an anime plot. Yeah. I mean, no shade thrown at it. It's totally fine and it's fun to watch in speedruns. So I think that the nanovirus is a solid addition to the game, but it ain't biology. So, okay. Do you find the diseases realistic in the cure mode? The cure mode will be like an entire thing on its own. I don't know if I'll actually tackle it because I'm not an epidemiologist or a doctor, so I can't really say too much about cures. I'm more of a little critter kind of guy. Okay. Let's talk about the bioweapon. This is fun. I like the bioweapon. So we'll talk about the bioweapon. We'll pull this up. So, let's just take a look at the language of the bioweapon. So the bioweapon is a plague of indiscriminate origin. The image for the bioweapon is very vague. I think artistically so, because who knows what this thing actually is. But I think as a couple cellmates had mentioned earlier, it does seem to resemble a virus. And so the bioweapon is actually something that can be, it like naturally accumulates lethality over time, but it's also very easily discovered. And so the like, gimmick, I think, of the game mode is to try to get the bioweapon to spread as fast as possible without being detected. And then at like the end game, you can turn on the like annihilate gene and it'll just like gib all of humanity. It's an interesting sci-fi like idea, but I want to like dig into some of the technobabble located in the text of the description. So there's a tree called gene compression, which just, I think that, you know, shout out to my programmers in the chat. You could argue that DNA could be compressed if you are thinking about DNA like data. I am not too familiar with how that would work, but I could see that happening, although that would have nothing to do with extending the delay between lethality increments. So I came up with something that might be a little bit like, you know, I would try. I mean, yeah, Aeon, you're right. It's like 100% sci-fi. But I mean, like, if Endemic wants to hire me to write for them so that they can make their game sound a little more official, I'm very happy to do so, you know, for the right price. But yeah. So preventing lethality from evolving, you could just say something like high fidelity polymerase. So if viruses have low fidelity polymerase to acquire mutations, a bioweapon could have a high fidelity polymerase. So basically stuff trying to prevent mutations from happening. I think that's very well in this good in the spirit of gene compression, you know, the actual in-game ability. DNA is highly compressible. Limited alphabet makes repeating patterns very highly likely, but I'm not sure the compressed form would be functional. Yeah, that's true. I mean, more juice, you know, it brings me back to my quarter semester of bioinformatics. I did like what I learned, but I couldn't make it work in my brain. Yeah, gene.zip, exactly. Yeah, that part I think of bioinformatics is really cool. I didn't have the talent, I think, to dive deeper into that, because you can also treat, you know, the DNA bases as code, right? Somebody might want to fact check me on that in terms of like, if we have bits that are either 0 or 1 in DNA, you have more than that. I'll just say that as a basis for programming, which I think is kind of interesting. Although I'm not really well versed enough in this kind of stuff to actually know what effect that would have, but it's fun to think about. What if a disease could turn one cell into another cell, like a blood cell into a liver tissue cell? So there are cells that are... So that would be tough. Like, we can do that in the lab, kind of. So there are scientists who research cell fate, which I referred to in one of my debut shorts, actually. The fact that every cell has programming within it that determines what they will eventually become, and it is very, 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 very difficult to make one cell into another cell. It requires a lot of different things to like get a cell to travel back in time, essentially. So whether or not a virus could do that, not entirely sure. Although I think something much more feasible for a pathogen to influence would be to take something like a stem cell, something that is relatively less differentiated and having a more open cell destiny, you can say, and maybe influencing that cell to become something different. That might be something interesting to look into. Hey, look, this polymerase paid for a WinRAR license. True, so true. Yeah. Okay. Bacteria and fungi are living things and viruses aren't, but what about prions? Are they living or not like viruses? So prions, this is a great debate that biologists have all the time in terms of whether or not viruses are alive. At this point, I think that the distinction between alive and not alive is less interesting than I think it was before, because like viruses can do a lot of things that living things can do, and alive versus not alive is kind of just a label for us humans to describe things as. You could describe every living being as just like a self-autonomous machine that can replicate, and that would lump us in with viruses, right? But if we were to like have a spectrum of life, viruses would be at the end of what we would consider living, and then prions would be, I think, not even on that spectrum, because prions can't replicate themselves. They can force other prions to become misfolded, but they themselves cannot replicate without the help of other components, we'll say. Although viruses can't do that either, but, you know, again, it's complicated. I imagine something, you know what, sorry, I'm getting so lost in chat. Let me finish the bioweapon, and then I will hop back into the conversation you're all having in chat, because I'm loving where that's going. So yeah, gene compression, it's kind of programming techno babble. Nucleic acid neutralization, I love that. That's so funny. Again, because we know that the backbone of DNA is charged, and so I don't know, what is this saying, that we're causing DNA to lose its charge to become electrically neutral? Like, what would that even do? If it's not ionized, will it like crash out of solution? Are we like ruining DNA by neutralizing them? I mean, I guess it's nucleic acid neutralization and not DNA proper, but I just, that's technobabble on top of technobabble. I love it so much. Deactivated modified genes. I mean, again, this is like sci-fi, right? And then unlock annihilate gene. Who knows what that annihilate gene is, but yeah, so there's some external control that scientists can have over whether or not a gene is expressed, whether or not it can be done over a long distance through many different physical barriers is another story. As of right now, I don't think it can be. So rest assured, I don't think the bioweapon will come for any of you. So I don't think it's anybody's surprise that I'm putting bioweapon in the technobabble tier. Okay. With that in mind, I'm going to close the poll on y'all's favorite of these, and we have prion. Yeah, I'm not surprised. Prion is definitely very, very popular. So let's set up. Can anyone remind me what the winners are for the other poll? And I'll set the final like Plague Inc. Tournament while I talk about some of the symptoms in Plague Inc. I'd imagine something that could change a cell into another cell would need to contain all the genetic context between the two cells and would have DNA payload at least as large or larger than both. Yeah, so I mostly agree with you, Morcell, in the sense that, like, it would have to be a pretty complicated set of things. If we can't do it on a bench, it's hard to imagine. I'm not saying that it's impossible, but it would be hard to imagine a pathogen doing it if we can't even do it on the benchtop with purified components. Whether or not you need a ton of DNA to do this is a different story, because, you know, I would love to do developmental biology one day on the channel, but if you hit a cell early enough in their like cell destiny, like a relatively young stem cell, then it doesn't take that many components, from what I remember about dev bio, to force them down one route of differentiation. But the context of those cells is going to be highly, highly specific. So it'd be very difficult, I think, for a pathogen to do. Virus 1 and bacteria 2. So then it'll be virus, prion, and nanovirus for this next poll. So I'm setting up the poll now. Favorite standard plague. Virus, bacteria. Then we'll do nanovirus and prion. Ah, I see, Margies. You're talking about transforming. Yeah, I guess I think about differentiation as being like transforming being like reverse differentiation. I kind of wrote off the transforming thing to begin with just because I am, from my knowledge, transforming is so difficult that it's more likely that you can influence differentiation, that you can influence post-mitotic terminally differentiated cells. So cells who's had their fate already determined. Although they can, but I don't have a citation for that, although I do believe that that is possible. Oh, fungus and virus. Oh, which one is it? I can do another round at the end if, because I don't think I included fungi on the other poll. That's okay. Well, the winner of this poll will fight fungi in the end. Okay. All right. Hopefully y'all having a good time. I love talking about biology. Biology is so cool. I want to talk a little bit about the symptoms. So in these standard plagues, a lot of them will share a similar symptom pool. And originally I wasn't going to cover this, but when I read the flavor text for some of these symptoms, I couldn't help myself. I thought that they deserved to be talked about. So what do we have here? Oh, so that's bioweapon. Let's remove this. Hypersensitivity. Okay. Let's take this here and blow this up. So hypersensitivity is one of the symptoms in the game. And it struck me as odd because it says increases likelihood of allergic reaction, which can distract the immune system. And I am, we'll say, I am unaware that I am unaware of a mechanism by which this is true. And the reason being that the immune system has different tools to react to different sources of immune stimuli. So, you know, us neutrophils, we're not necessarily going to be the cells that are recruited to fight an immune reaction, right? So those immune reactions to allergens are going to pull from other cells like eosinophils and mast cells. So I don't know that the immune system for pathogens would really be distracted, although I do have to admit that I don't know enough about allergies to confidently say that that's true. So I'll put hypersensitivity in the I guess tier. It's, if it is true biology, I don't think it's commonly known true biology. So that gets a I guess. Is it legal to experiment on random diseases at your house? I'm not qualified to answer that question, but actually, More Juice had suggested something to cover something in that realm of backyard experimentation in the members only suggestion box, which you can contribute if you join as a member. I have a thing called like the Learner Challenge where members can basically guide the kind of content, the learning content that I make. So if there's something that you really want me to cover, you can put it on a list and I'll get to it. So More Juice, I don't want to like spoil the exact topic because I think it's really fun, but it does have something to do with backyard experimentation. Okay. Next up, hemophilia. We'll pull that one up. So hemophilia, according to the game text, immune system produces inhibitors that destroy factor 8, preventing blood clotting. So this is really, really interesting because I, again, not a doctor, don't know a whole lot about the clotting cascade. And to me, this sounded like technobabble because, well, how could it not, right? Immune system produces inhibitors that destroy factor 8. I've never heard of any pathogen doing that. But when I looked into it, I actually read that certain types of infection can cause the immune system to develop neutralizing antibodies to clotting factors, meaning that, like, because my impression was that diseases that caused issues with clotting were directly, like, causing problems with clotting, like producing proteins that would inhibit clotting. But for some infections, this appears to be a actual thing that can happen, which blew my mind. So again, when I learned something that I didn't know before, and like especially when it changes my mind about something, that automatically gets it into the superb tier. So that's really fun and exciting. Not exciting, I mean that kind of sucks, but it's an interesting strategy that pathogens have evolved to take advantage of, that triggering the immune system to attack its own clotting factors. That's pretty wild. This is realish at least. Factor 8 is in the clotting cascade. Yeah. I was really surprised. I don't know anything about clotting, but I think it's really cool. Someone did their research, huh? Well, yeah. I mean, yeah. You know, yeah. I don't know if you're referring to me or Plague Inc., but I'll take credit for that. Snake venom does the same thing depending on the snake. Interesting. Like it produces an immune reaction that causes the immune system to destroy clotting factor. That's pretty cool. I would love to do a toxin video at some point. Another field that I'm not very well acquainted with, but I did work quite a bit with tetrodotoxin as a graduate student. Tetrodotoxin, for those of you who don't know, is a neurotoxin isolated from pufferfish. It's the thing that makes fugu a less than completely safe choice to eat, and it is one of the most potent neurotoxins on the planet. You basically shut down neuron communication, and you're completely aware the entire time as your nervous system shuts down. Very scary, but very easy to avoid. Just don't eat fugu. That's all. Can you rate PAX 12 too? Yeah, for those of you who don't know, PAX 12 is the default name for a pathogen when you load into Plague Inc. PAX 12 is superb, I think. Superb pathogen. Is the point that preventing blood clots allows the infection to spread faster in the body? I don't actually know what evolutionary advantage preventing clotting would have for a pathogen. So that'd be interesting to look at in the future. Very interesting. Very interesting. Have I heard of the hypothesized link between MS and mono? No, I have not. Although I am aware that there are viral hypotheses for nervous system disorders. Not nervous system disorders, but what do I even call them? Diseases of the nervous system? Maybe that's the right term. Not entirely sure. There's a protein on the virus that is identical to a protein on the myelin sheath that the immune system can get confused with and start the process of MS. Interesting. Yeah, I mean, I think it's mechanisms like that that make Plague Inc. a bit... Like, if I were to be on the consulting board, there are wildly interesting mechanisms by which things occur. Although maybe it's a deliberate choice by endemic to not have it be too real, because you can imagine that there might be the game might not be as fun if you're like that explicit with real disease, because real disease affects real people. I mean, you know, in stream we can kind of laugh and joke, but these are real things that real people go through, and it's important that we keep that in mind. Yeah, which is why like I try to be pretty careful about keeping these streams like as straightforwardly about biology as possible. Botox is a neurotoxin too. That is true. Botox botulinum toxin, I think, right? Is the full name. That would be an interesting one to do too. I love like I think neurotoxins are really, really interesting. I would love to do a stream just on neurotoxins actually, because those are the ones that I'm most familiar with. Cells at Work is trash. I highly disagree. I very much love Cells at Work. I think it's an amazing, amazing anime. And I had some similar ideas of making my own at about the time that Cells at Work came out. And that evolved to be this channel, actually. Welcome, Female Thunderbirds Nerd Gaming. Welcome to the Plague Inc. stream. If you go back to the second, I'm not sure though, in the trailer they kind of show the real PAX 12. Really? I should look into that. Would you ever do analysis on fictional things like black light or red light viruses, or maybe something from Resident Evil? I'm happy you asked, because I think like it's either the first or second short on my channel. No, wait, no. Well, yeah. Progenitor virus is Resident Evil, right? I think I did a short on the progenitor virus at the beginning of my channel. I wanted to do another video about Resident Evil, but it became too complicated. So, maybe one day when I'm done with my highest priority videos, I could revisit Resident Evil, because there's more to talk about in Resident Evil. It started with a short, and then I was like, oh, I gotta have to explain so much about genetics for that to actually work. But keep that in mind, I started this channel doing video game biology. I'm not planning to abandon that. Just I have a lot more long videos that I want to do first. Did you see Cells at Work Black? I read Cells at Work Black. Oh, Cells at Work Black is so good. I didn't watch the anime though. But the art in Cells at Work Black, there are frames that are so emotional for me, which is really like, I don't know. It's a very like geeky thing to say that like, oh, this like anthropomorphic T-cell that's like in tears is making me cry. But like the art is so good. It really, really makes you feel like it goes so hard. It makes you feel for, and it's like kind of my channel too, right? Like every entity in biology can be humanized. It's not necessarily the best way to learn biology for your classes, but in terms of keeping your interest and like evoking your imagination, there's nothing wrong with personifying biology in that way. So I highly recommend that everybody go read/watch Cells at Work Black. Imagine finding the neutrophil with cap and wielding a knife. I do not want to get demonetized. Forgive me, isn't anthrax a real-life bioweapon? I heard in the episode of Simpsons when we were introduced to Krusty's daughter. I don't know if anthrax was a... I don't know if it was a hoax or not. It's been a while since the anthrax scare, and I was too young to actually understand what was happening back then. It might be interesting to look at again in the future. I couldn't imagine the platelets suffering in Cells at Work Black, so I avoided it. Oh, but the thing about the T cells, it's one of my strongest like visual memories. I'm getting emotional just thinking about that frame. I thought of a concept for a virus-like structure sitting in the inactive portion of cell such DNA that can express itself by infecting or damaging the host cell, repairing it. Inactive portion. I can't think of a virus that hangs out in the nucleus because I'm not a virologist. So I'm wondering how feasible that would be in terms of like hanging out in the nucleus. Well, there's also the fact that once a virus gets in, it's not really, it doesn't really have its shape as a virus anymore. It's by that time it's its DNA or its genetic material. Uh, ba, ba, ba. So I'm reading chat right now. All right, so let's get into the last symptom and finally close out this Plague Inc. stream. So this is immune suppression. So this is the last of the symptoms that I want to talk about. So we'll bring that up. Immune suppression. So if you want to summarize my channel, at least by view count of what my channel is supposed to be, it would probably be this, right? I like telling the stories of immune evasion. I think immune evasion is the more common term with regards to understanding the relationship pathogens have with the immune system. But I think suppression is completely fine. It doesn't really ring as technobabble to me. So this one says pathogens attached to lymphocytes, so cells of the immune system, like me, suppressing the immune system and allowing significantly greater freedom of mutation can be fatal. So this one's a little funky. Pathogens attached to lymphocytes. As of right now, I am unaware of a pathogen that just flat out attaches itself to a lymphocyte. Maybe there's one out there. I don't know of one. Maybe I'll learn of one in the future. But yeah, just like a cute little pathogen just hugging a lymphocyte, like those parasites that like hang out around sharks. Not parasites, mutualist symbiotes or whatever you want to call them. HIV. Does HIV hang out on the outside of cells? Like do the capsids just like stick there? Because I had imagined that the DNA would go inside the cell, right? Attaching, I imagine, as being like sticking to the outside. Yeah. So, the connection between this and the game mechanic, we're talking about increasing the freedom of mutation. In an indirect way, I guess, if you have immune evasion and you're able to survive longer in your host, then that leads to probably more mutation, but it's not really a direct connection, right? Like you have more freedom to mutate because you survive longer. So it's an okay kind of explanation, I think, although I wouldn't really word it that way. And if anything, I would think that immune evasion would make something like a cure harder to do or make severity increased. It doesn't really ring as solid as some of these other things have been ringing as. So I'm going to actually put immune suppressor into the suppression to the I guess category. I think I'm happy with where that belongs. Okay. I'm going to have a look at the poll now and see what round 3 looks like. So we are, oh my god, we're tied between viruses and prions, it looks like. That's incredible. 32% virus, 32% prion. Oh, 31, I guess. So I'm going to do a third poll, and this time we'll actually loop in the rightful winner of that last poll. So we'll do virus, Final Plague Inc. Tournament. We'll do virus, and we'll hit it with a prion, and we will also do fungi, right? Fungi was the other winner, right? Fungi. Oh, I just scrolled up and I was like, sorry, I didn't mean that the anthrax wasn't real. I meant that I was wondering whether or not the whole thing about anthrax being mailed was a hoax. I wasn't entirely sure. Fungi. Okay, yeah, it is fungi. Okay, great. Fungi. Okay. Something that I haven't thought of. Do viruses have species? So yeah, that's why like I don't think that the line between viruses and all the other things that do similar things to viruses is really, it's not even really worth arguing to me anymore, because you can trace their evolutionary relationships with one another too, right? They have genetic codes, so you can compare the genetic code and sort them by lineage. So they do have groups. I don't actually know if they're called species. I don't know if Evian can elaborate on that. Our resident virologist in chat, if y'all are still around. Anthrax was for sure mailed out. It was a case study in response to my old job. Oh yeah. Yeah, okay. So it wasn't, maybe I was coping when I was young and just hoping that it wasn't real because I was such a anxious little child. Virus classification is a big thing. You can search ICTV for that. Do they call viruses by species, or is that reserved for what people commonly call living? I think that would be interesting to sort out. All right, that's like it for the tier list. I'll stay around and hang out, but I hope that I hope that this plaguing stream was everything that people had hoped it would be, or at least found it interesting. Fi, is it true that there aren't white blood cells in the brain? So there's a blood-brain barrier, and if you find cells of the immune system in the brain, then usually it means that something is wrong. So that doesn't mean that there are not white blood cells in the brain, but if you find them there, it's trouble. That doesn't mean that the immune system isn't active in the brain, because the brain has its own immune system warrior, I guess you could call it. But yeah, if you find neutrophils, T cells, macrophages, if they're hanging out in the brain, something is wrong. That's not supposed to happen. All right, thank you for coming to the stream, Mitochondrian. Have fun swimming. Because I got kind of curious, because a lot of viri get code names, kind of like proteins or molecules. Yeah, I mean, they have to be grouped because they share like similarities. I think what's fun is that if you go to, what's it called? God, I can't remember. There's a website where you can look at the tree of life, and like the entire tree of life, and see all of our evolutionary lineages. I don't remember what the website is called, but you can see sort of the different classifications of not only like microbes, but also across entire species. And the classification for like archaea, as I've been reading a lot lately, pretty neat. They've got like acronyms for species names, which I think is not as common when we think about higher organisms. I gotta say, a stream where I can let my disease nerd fly freely is super nice. Yeah. I mean, it's such a sensitive topic in the sense where you don't want to make light of the diseases, but at the same time, like, the people that devote their lives to studying these things, they are not always just motivated out of the goodness of their heart, right? You want like nerdy disease people to study disease because they're going to be good at studying disease. So I think it's fun if it helps elicit joy in you to learn about biology. That's all I really want. Mostly, virus classification is quite hard. The sequence conservation is low, so people frequently use structural conservation of a few conserved proteins. Oh, that's interesting. So that's actually kind of more like classical biology, right? If we're talking about morphology of species, before genetic sequencing allowed us to trace genetic lineages, it was a lot of naturalists looking at different animals' bodies and being like, yeah, these are probably more closely related than these two. But it turns out in the advent of sequencing that those relationships are not necessarily as true. There's one called Candidatus Hilaricaea Toa Asgard Archaea. That's so cool. Yeah, dude, Archaea are cool. There's not a whole lot known about Archaea for reasons that I will get to in my Archaea video, which you should all subscribe and hit the notification bell because YouTube is YouTube. And, you know, if you're not on your subscriptions page, you could miss videos that I put out. So, you know, I'd hate to be every YouTuber ever, but please hit that notification bell. I guess I never really thought about what different staphylococci I have in common. I just kind of accepted them as relatives. Yeah, I think that genealogical relationships between species is like a level of hardcore that I could not get into. I just accept it. I just say yes. Yes, and. Before trigonometry killed me in college, I wanted to go into BSL-4 research. I wanted to too as a kid because I had not only this great fear of disease, but also a great curiosity about them. But when I took a couple of semesters of lab in undergrad and then in graduate research, I was like, oh, I would absolutely like die. You have to be quite careful. You have to be quite careful when you work in a BSL-4. You know, we're talking like hazmat suit, get a little on you and you die, like level of carefulness that I just don't have. I told the story in a previous stream, but one time I, you know, and it's a lab mistake that I'll never forget. And if y'all are thinking about getting into chemistry, never do this, right? You're supposed to neutralize your acids and bases before you pour them down the sink. I was lazy. I just diluted mine by a lot. I added a whole bunch of water to hydrochloric acid, and then I poured that down the sink. And then later on, when I was disinfecting some cells, I poured the bleach that I used to disinfect the cells down the sink, and hydrochloric acid plus bleach equals chlorine, and I ended up gassing out my entire lab. So I do not belong in a BSL-4 lab for sure. Yeah. Considered already done. Thank you so much. Hit the sub button or you get PAX 12 true. That is not a threat, though. Legally, I am required to ensure the audience that is not a threat. What type of plague is the Black Death? So I believe the Black Death, I'm just going to look it up real quick because it's be real embarrassing if I got it wrong. Yersinia pestis, what is it called? Bacteria, yeah. Yersinia pestis. Yeah. Definitely something that I will... you caught me looking it up on Wikipedia. But yes, just wanted to make sure that is correct. You chlorine gassed your lab? Yes, I absolutely did. My lab had to evacuate. Thankfully, the whole building did not have to evacuate, because I would never live that down. It was just my lab. Our lab has pretty good ventilation. They just said, evacuate the space, wait for like 15, 30 minutes because there's a lot of air change in biology labs. Because both the bleach and the hydrochloric acid were both dilute, it means that I didn't create enough chlorine gas to like kill people. Like, it wasn't apocalyptic, but if I hadn't diluted the components, that would have been really bad. That would have been, I would never live it down level of bad. There's a channel, probably a bunch actually, that does readings of viewer shared chem lab incidents. I don't know if I want to share mine. Mine was pretty bad. Although, yeah, I was like one step away from being one of these cautionary tales that universities teach you when you do laboratory research. So for those of you who have not done like science lab before you're allowed to like touch any chemical or work in a lab, you typically get what's known as safety trainings, right? And every university has their own safety training, and it's the most horrifying thing you've ever seen. They will tell real stories from real labs at your university of terrible things that have happened to real people, including, I can't remember, in my lab safety trainings, there was a figure of a centrifuge that was not balanced. No, it was I think it was both. A centrifuge that wasn't balanced that blew up. So for those of you who don't know, a centrifuge is essentially a machine that spins things really quickly, and it's really important that it be balanced. So if you load one tube on one side, you need to load another tube on the other side of the same weight. Because if you don't, then the rotor can become unbalanced, and if the rotor becomes unbalanced, that thing can launch itself like a missile. And so there was just this chaotic damage that the safety training showed us were just brutal. Like a missile had tore its way through multiple floors of the building. Same thing with compressed gas. So, you know, it's all fun and games when you watch a Nile Red video and he's got a whole thing of liquid nitrogen and he's pouring it out, and that's fun. But compressed gas is also extremely, extremely dangerous. If you cap compressed gas, then pressure builds up in whatever container that you have it in. And if for whatever reason the pressure can't hold, you end up with another ballistic missile type deal. So those are very, very, very scary. I threw a chlorinated rat at my research director. Is that a real one? That chemist, is that the channel? I'll check it out. Our TA said someone once put an acid in a full 5-liter base waste bin and things didn't go so well. Oh my gosh, that's an explosion waiting to happen. So yeah, acid-base reactions, exothermic, heat generating, that's very scary. Another story that we were told in biosafety training was, oh, I forget what the chemicals are called. Peroxides? I forget what they were called, but basically they're salts that can accumulate that are shock sensitive. So they're just, it's just like grains of sand that can explode if you move them too hard. Which is why they were like, you should always, always, always label your waste, you know, with the full chemical name, and you should do it in a way that is readable so that if you leave behind a waste bottle, the next person definitely knows what's inside, because it might be full of dust that can explode. So yeah, so definitely exercise caution. Hydrogen peroxide and acetone, TATP, those sound familiar. Yeah, it feels good to stream again. I wish I could stream more, but you know, I have like a full-time job, which has been quite busy recently. But the reason why I haven't been streaming, the reason why I didn't stream last week is because I've been cooking up this Archaea video. And I'm kind of curious as to what level of education you, or what level of baseline knowledge you all have about Archaea. Oh, yeah. Sorry, I forgot to do this poll. So let's look at the poll right now and see who the winner is. And oh my god, it's so close. Okay, the winner of the By the Neutrophil Plaguing Tournament is a freaking tie between viruses and prions. That's insane that's so close. That's amazing. But there was another poll that I wanted to do in terms of like content strategy, and I'm not saying that if chat votes on this that I'm going to go with it, but I kind of want to hear how this hits your ears, you know, speaking of prions, because one of the videos that I want to do in the summer when I have more time is like a very, very high effort prion video, but I kind of want to think about how to roll that video out. And we'll do a 1v1 on viruses versus prions after this, because I want to take note of this before I forget. So I want to get a sense for whether or not you would prefer a tight narrative video about prions, or if you want me to do a video where it's everything I know about prions and it doesn't really have like a video essay, one of those long ones. And I'll do that as a poll. That's actually a thing too, growing a little fungus ball in your heart. That is terrifying. That is terrifying. So this would still be long form content. So the tight narrative would be about 10 minutes, and it would be only the most interesting things that I know about, that I will have learned about prions. Whereas like everything I know about prions, I worry because it's harder to keep a tight narrative, and it's harder to keep visuals interesting if I continue to talk. So the reason why I like doing these 10-minute snappy videos is I can cram a lot of visual jokes in there if my script is short. But the longer that I talk, the less I can think about interesting things happening on screen, right? So the 5 bits are a really good excuse for me to just talk relatively unedited because I don't have to worry about visuals on screen. So yeah, I kind of like at the temperature if we want, or if we would enjoy like a more traditional video essay type thing where the on-screen visuals aren't as engaging. Aspergilliomas. Maybe a topic for a future video. Why not both? I mean, I guess we could do something like I did for rabies, where I initially do a tight narrative video and then do a 5-bit. I kind of do like that idea. Because I can keep the narrative really tight and the visuals interesting and be funny, because in the 5 bits, there's not really room for humor. But in a 5-bit, I could just talk about everything that I've learned without caring too much that everything makes narrative sense. So I think that's a decent strategy. Yeah. That's a good idea, Toxo. Or a bunch of tight narrative videos that all encompass everything Fies knows about prions. I worry that people will bore of prions. I mean, I know that like my core viewers will probably love the prion stuff. I'm less confident about the algorithm as a whole. Oh my gosh, thank you so much, Merkala Camstein. Please tell me if I've pronounced that correctly, but welcome to the Helper T cells. Welcome. Oh my gosh, let's freaking go. Welcome. Spam some 5 strongs. So at the Helper T Cell reward tier, you can instruct me or guide me towards topics. Unfortunately, the topic for April has been chosen already. But in the future, if there is something burning that you would like addressed, I'm very happy to, as long as it's somewhere in the realm of biology, sociology, and psychology. So there's something that I want to clarify when I talked about membership. I had called sociology and psychology easy topics. That's not really fair, I think, because I'm perpetuating the idea that those are easy disciplines, which they're not. It's just they're easier for me to get into because of something that I want to address called, what's it called? Bloom's taxonomy. So is anyone in chat aware of Bloom's taxonomy or knows what Bloom's taxonomy is? Because I don't want to perpetuate this like STEM elitism or like the quote unquote hard science elitism. So I want to address what I actually mean by easier. So I'll bring up a thing. How do I spell that? Okay, I'll type it in chat. Bloom's taxonomy. Your level of comprehension makes it easier to grasp and translate. Yes, so Reunite, you've got it. So I'm going to put this figure up. And this is a little bit of like teaching metas. So for those of you who don't know, like my job outside of, doing this as a hobby is I'm an actual biology instructor. So I think a lot about how people learn on a mechanistic level. So because I would like to have my teaching be informed by research, like evidence-based practices, because I just think that that's cool. So let me bring up Bloom's taxonomy so I can share that. So this is like a model, and I refer to it explicitly as a model because I couldn't say whether or not this is quote unquote true. Like, I don't know if this is how learning works, but it is a way to think about learning, that learning happens in these stages, we'll call them. So when you get into a topic, the like lowest form or the entry point is going to be your knowledge, right? So if you've heard of a prion, then you have knowledge of that prion. But like knowing that a prion is a protein that misfolds other proteins when itself is misfolded is comprehension of a subject, right? So these are stacks of layers of understanding. And so you get application, where is the application level is where you can start answering questions about the term. So like if you were to learn about prions, if you can answer things about prions, then you have a level of application. And then higher than that, you get like analysis, synthesis, and evaluation, which is talking about like an undergrad to grad school to expertise level of understanding, where you can take data in the field and not only comprehend and understand what the research is saying, but also then criticize it or evaluate it for truthfulness. And so it's very difficult to get all the way to the evaluation stage of information, right? My goal for the channel is to take you from knowledge and to bring you as far as I can on this ladder, because I think those are where the most interesting things are. But not in a way that I am serious, in the sense that if I wanted to teach, I would do something like Khan Academy, for example. This is partly to entertain, right? But what I mean by psychology and sociology being quote unquote easier for me to understand, it's more like the level to like get your foot in the door. You don't have to learn all that much. So like to get into psychology as a field, like just to like have a basic understanding of what's going on, you don't have to read quite as many things. Whereas in biology, right, before you can learn all the fun stuff that happens in a cell, you have to learn about the membrane, you have to learn about chemistry, you have to learn about all these other things. But that doesn't mean that psychology or sociology are any easier as disciplines in the sense of being able to affect having actual meaningful discourse, right? Like, I'm not saying that it's easier to be an expert in psychology or an expert in sociology, because that requires reading everything in your field. So I just wanted to point that out. But also, I think it's just kind of neat to share a little bit of learning meta, if you weren't aware. And if you're a student, you can definitely like apply this to your own life, I guess. I mean, you don't have to be a student. This is just in general, right? I don't know. I think it's interesting. All right. Yeah. I'm getting kind of hungry, y'all. Looking to do some phagocytosis. I think we're probably gonna call the stream there. I had a great time. I hope you all had a fun time learning about what I think about Plague Inc. Eat some bacteria. Will do. I don't know if I will stream next week. It would be tough to stream next week. I'll just put it that way, because I put in a couple of hours of prep into these kinds of streams. If I do stream, it might be a just chatting thing where I don't have to prep that much. But if I don't stream next week, it won't be a surprise. When I was in high school, I used to love science. I honestly didn't know you were a biology teacher. Thank you for explaining these Plague Inc. in a fun visual way. I'm autistic, so thanks. No problem. Yeah, like my number one passion, even beyond biology, is to share my excitement for things that I find exciting. And if I had specialized in any other subject, I would probably at the core be doing something similar, but maybe for a different subject, because when people find the things that I find cool, that's like the best feeling in the world. Meta learning. Be a meta slave. True, I am a slave to the learning meta. One day I might want to do a stream on learning meta if those papers are free access, or open access, I should say. Just because it's kind of, at least for me, it's kind of interesting to think how researchers think about people learning and how we can measure that. It's kind of neat. The stream was really good. I really love Plague Inc. and I'm going to play it after stream. Please do so. If you haven't watched Easy Speezy speedruns of Plague Inc., Easy Speezy being a very big speedrunning YouTuber, definitely check that out. It's pretty mind-breaking how speedrunners have just completely blown this game open. All right, with that, I think I'm going to call it a stream. Thank you all for coming. I hope you had a lot of fun. I will see you in a week or two. As a teaser for you, all of you who are still here, April/May is going to be a big month for Find the Nutriflow channel. So, keep your eyes peeled for some exciting things that this cell has been cooking up in the background. And with that, bye y'all. Goodbye. Bye.